Paper is non-invasive assessment of body conditions and stress hormone levels in African elephants roaming in
Fynbos vegetation. Yeah, thank you. Okay, so good afternoon, everyone. I'm Elisabetta, and I'm from Italy, but I'm an MSc candidate from the University of Pretoria. But today I'm going to introduce you my research from last year, my honors, always from the University of Pretoria, on elephants in Fynbos vegetation. So as probably many of you know, the Western Cape province of South Africa hosts the Cape Floristic region, which is an important hotspot of endemic biodiversity. In this region, we can find the Fynbos vegetation, which is characteristic of the Fynbos biome found in the area. Here you have some of the most important species, such as Protesia, Ristonicia, Fabaceae, and this is recognized worldwide as one of the most important global spots of biodiversity. The Fynbos includes, mainly succulent shrubs and some evergreen small shrubs. Okay, so if you look at the western landscape of South Africa, especially on the south, you will see that it is dominated mainly by these small shrubs, not trees. But why? Because the soil is poor in nutrients, and so this is also reflected on the vegetation, of course, which in turn will be poor in copper, calcium, zinc, phosphorus, and magnesium. So it's been questioned whether this type of vegetation is actually able to sustain large herbivore populations. We know that a balanced intake of food is very important for some physiological and psychological processes. And so in some cases, artificial supplementation might be needed. Okay, so during the mid-17th century, the European settlements in the area became permanent, so these led to a lot of anthropogenic pressure, and it also led to some local extinction of some large herbivores, among which the elephants. So the less remaining populations were forced to utilize less accessible areas, and this means that these animals once were free to roam from different type of environments, eating different type of vegetation types, and now they had to adapt to utilize only certain plant species. We know that elephants are quite adaptable species. In fact, we can find them from the deserts to the savanna areas to the forests. And this is why they are mixed feeders. So they can feed on anything from grasses to herbs to woody plants. They are on-grass fermenters, so we know that they're not ruminants. So they have this large cecum where the microbial fermentation takes place. And this is why the nutrients absorption in elephants is not as efficient as that of the ruminants. In fact, they have to consume large amounts of food every day, around 450 kilograms, but they can only absorb 44% of this. Okay, so after this long introduction, this is our research site where the research took place in this type of environment. This is the Gondwana Private Game Reserve, and it is situated in a Fynbos vegetation area. So we had to assess the body conditions and the stress hormone levels of a herd of 13 elephants. This herd was composed by 2 bulls, 4 adult females, and 6 juveniles, and 1 cow. And we monitored them in two periods, April and June 2018, that correspond to the early and the late wet season. Okay, so the aims of the study were to determine the individual body condition score, to determine individual fecal glucocorticoid metabolite concentrations, which can be a difficult phrase, but basically means that we wanted to assess whether these animals were stressed by measuring levels of stress hormones in their feces. And we also wanted to examine variation in individual BCS concentrations, and F2CM concentrations during the early and the late wet season, And in the end, we wanted to examine whether there was a possible correlation between individual BCS and FGCM concentrations. Okay, so let's start from the beginning. As you probably know, a useful tool to identify elephants is to draw particular features on these ID kits, because at first sight they might look all the same. This is an ID kit from Bonnie. She was one of the oldest females. She was 34 at that time. And as you can see, she has this characteristic right tusk that is bent backward. And here you can see Bonnie in all her beauty with that particular tusk. Okay, and well, some other times you can't rely on tusks, so you have to rely on something else, and notches on the ears are quite important to be able to distinguish animals. So in this case, this is Big Grip. We called her Big Grip because of that big grip on her left ear. So we know we didn't have a lot of invention with the names. And these are other ID kits that we just made. Okay, so one of the aims of this research was to assess elephant body conditions, because it is known that food availability and environmental characteristics can influence body conditions and can also cause stress. So, as you probably know, this BCS method is being originally used in livestock industry to assess body conditions, generally to improve productivity. But it's also been used in the last decade to investigate general health conditions in free-ranging and captive wildlife. So we used this method from Morfeld and colleagues from 2014, who developed one of the first visual methods for scoring body conditions in African elephants. And this method is based on a scoring system ranging from 1, which corresponds to an emaciated state, to 5, that corresponds to an obese state. And this scoring system is needed to visually assess standardized pictures of each individual by analyzing some anatomical key areas. And you can see the key areas in this picture, for example, the ribs or the pelvic bone or the vertebral ridge of the backbone or the lumbar depression. Okay, and this is just a BCS flowchart that we use it as an intuitive guide to lead the assessor in evaluating body conditions of the elephants. You can see the lower scores, which are 1 and 2, are indicators of poor health conditions, some low reproduction rate, and in general, malnutrition. While the highest score, which is 5, is associated with reproductive failures, obesity, as we said, diabetes, and other pathologies. So we took at least 3 relevant pictures for each individual for each site visit, which means that we had to take a picture from a side, 1 from the rear, and 1 from 45 degrees angle. And then these pictures were assessed by 2 different assessors to address inter-observer variability. This is just an example. This again is Big Rib. And as you can see, the ribs are not visible, the pelvic bone is visible, but is not too prominent, as well as the backbone. So based on the flow chart that I just showed you, this elephant should have a BCS of about 3. Okay, so the second aim was to assess stress. But what is stress? So stress is a response to a stimulus that threatens the homeostasis of an individual. Basically, there can be internal or external factors that can act as stressors. For external factors, we can think about challenging weather conditions, availability or quality of food, or intra-specific competition, for example. And for internal factors, we can think about injuries or diseases. So these factors, as I said, are called stressors, and when an organism recognizes a stressor, this triggers into the body a cascade of different physiological and behavioral responses, which activate a response, trying to restore the homeostasis. So in such a situation, the neuroendocrine system, which is dependent on the central nervous system in the brain, activates, among the others, the hypothalamic pituitary adrenal axis, which releases into the blood species-specific hormones called glucocorticoids. So these glucocorticoids are the keys of our study. These hormones, in the end, will be excreted through feces and urine. Okay, so when a chronic stress situation occurs over a prolonged period of time, so when you have that cascade of effects over a prolonged period of time, these can cause a lot of problems in a body, such as biological and physiological alterations. For example, different pathologies can occur, as well as the suppression of the immune function, so the animal will become more susceptible to be infested by internal or external parasites, for example. And of course also reproduction failures, as well as behavioral and conditional problems can arise. Okay, so we here listed just some of the possible factors that can cause a rise in FGCM concentrations for African elephants. As you can see, availability and quality of food can cause stress, competition for females, for example, in males, disease or injury, as we said. And then mast. It's been questioned whether mast can actually cause stress in the old bulls. But it's been proved by a previous study that this sexual condition is not associated with significant levels of FGCM concentrations. So mast is not really associated with stress. Okay, so how do we assess stress hormone levels? As I mentioned previously, these stress hormones called glucocorticoids get excreted through the feces. So by collecting and analyzing dung, the hormones can be used as a physiological indicator of the stress condition of an individual. So you know that when we work with animals in the wild, a lot of different challenging conditions can happen. So assessing stress hormone levels can be done in a non-invasive way, as we have done. So yeah, this method of collection is also viable, as basically the animal is not disturbed or affected by the collection, and these in turn won't be reflected in your last analysis results, which is very important, because if you cause stress and then that stress is reflected in your last lab analysis, then you are the stressor, so what's the point? Okay, and of course there is also a method of collection, because samples should be collected within an hour from defecation, and the hormone composition tend to be influenced by environmental conditions such as bacteria, such as temperature or moisture in the area, and also to avoid sample contaminations from urine or soil, because you also don't want that. You should open the pile of dung and collect from the center of the bolus of the pile. And also, of course, you have to use latex gloves, like in the pictures there, because you don't want to have any cross-contamination with some human hormones. Okay, and then you have to put the samples into a plastic container and then into a cooler box with ice, and then these samples have to be frozen at -20 degrees Celsius until lab analysis will be performed. Okay, so our analyses were performed at the Endocrine Research Laboratory of the University of Pretoria, where the samples were prepared through pulverization, weighted, and then the fecal steroids were extracted. And then the hormone concentrations were measured. So we collected a total of 42 fecal samples during the field work, and also a total of 150 pictures from all the individuals were taken. Okay, you have your elephant ID, and then the BCS for April and June, and then the median FGCM again for April and June. Let's say if we look at the BCS, we can see that basically almost all the elephants had a BCS between 2.5 and 3. which seems acceptable. Only one, the bully, showed a BCS of 2 for both the study periods, and also his stress hormone levels were quite high in June. And we tried to understand what was going on with him. Okay, so our results. As you can see on the graph on the y-axis, the individual FGCM concentrations, and on the x-axis, you have the month sampling periods. We had the chance to use some samples taken from another researcher, Dr. Gabi Teren, that previously studied these animals in December 2017. So the graph showed that the overall median FGCM levels were 34% higher in June 2018 compared to December 17 and April 2018, but the difference was not statistically significant. And the comparison of median FGCM concentrations for the same individuals in the 3 different sampling periods could not be conducted due to a low sample size. So there were only 4 overlaps individual samples, so we could not conduct the comparison. Okay, and then considering only the 2 sampling periods of the early and the late wet season, the individual median FGCM concentrations did not differ significantly between April and June. It's interesting here to notice how the highest and the lowest FGCM values during June are from the only 2 bulls, and that point up there is bully. As I said, yeah, we will talk about him just now. So on the last graph, we can see that the correlation between individual elephant FGCM concentration and BCS, they did not correlate at any of the 2 months of the 2 study periods. So in conclusion, the majority of the elephants had a BCS between 2.5 and 3, which seems acceptable. So there was no need for supplementing their food at this stage. And then determined FGCM concentrations were comparable to those of other non-stressed elephants from previous studies, and then our results also did not underline any correlation between seasonal BCS and FGCM concentrations. Regarding bully, our bull, so we really tried to look for any sign of injury or distress or disease, but from an external examination, we were not able to see anything. So we recommended the reserve to monitor the bull over a longer period of time to check for some sign of injury or disease, and if this body condition should continue, should persist, then we recommended to call in a vet to check his physiological conditions, because there might be some problem that a vet could solve. Okay, so the ability of this species to overcome environmental stress and survive in Fynbos vegetation has not yet been sufficiently studied. So further studies on the vegetation type these elephants feed on would actually be useful to investigate their real ability to survive in such a unique environment. So we believe that integrating non-invasive methods, like the ones that we just used, to assess glucocorticoid metabolites levels in body conditions can provide wildlife managers with a solid tool to monitor elephant conditions, living in different type of environment, not just this one, making weighted management decisions for the wellness of the animals. Okay, so I just have a few acknowledgements. My professor, André Hansvin from the Mammal Research Institute of the University of Pretoria, Dr. Gabi Teren from Wildlife and Ecological Investments, the Endocrine Research Laboratory from the University of Pretoria, the Mammal Research Institute from the University of Pretoria, and Eugene Marais Chairs for Wildlife Management, again from the University of Pretoria, and thank you all for your attention. Thank you.